Alright, so the next thing we're going to talk about is we're going to take a look at the ear, and really this first one here probably a little bit more about ear structure. The next one we'll get into a little bit more of the physiology of how this is working. So really when people look at the anatomy of the ear, they kind of divide it into three main sections. You have your external ear, your middle ear, and then your inner ear. So the external ear is really, when we think about what is visibly, if we told like a 2-year-old to a 3-year-old point to your ear, they'd go something like that. That is what we refer to as the auricle or pinna. This is that pretty much structure that is going to help direct those sound waves into our ear canal, which is known as the external auditory meatus. This is going to be lined with earwax glands, those ceruminous glands, and is going to take those sound waves to the tympanic membrane. The tympanic membrane, a lot of times known as the eardrum, is what's going to vibrate and then transmit those vibrations through the middle ear to that inner ear. So the middle ear, again, is an air-filled space. It's obviously in the middle. Really a combination of a few things here, but it starts at that tympanic membrane, connected to that tympanic membrane to mechanically move those vibrations through are the auditory ossicles. These are the smallest bones in the body, sometimes nicknamed the hammer, anvil, and stirrup. These are the malleus, incus, and stapes. That stapes connects to what is called the oval window, which is going to help move those sound waves from that middle ear through it to the actual inner ear and move the fluids that you'd find within the cochlea. So these are your auditory ossicles. Like I said, the malleus, incus, and stapes, or hammer, anvil, and stirrup. You can see why the stapes kind of gets the nickname stirrup. Honestly, the incus, you can see why it's nicknamed the anvil as well. Malleus, it doesn't look as much like a hammer, but if you have an anvil, you might as well have a hammer. So other parts of this middle ear are going to be what we refer to as the auditory or eustachian tube. Again, because we have air-filled cavity behind here, you are going to have atmospheric pressure on the outside of the ear here. You're going to also need, if this is going to vibrate freely, it needs the same pressure behind it. If there was a buildup of pressure in here, it would bulge this out. If there was a vacuum relative to the outside in here, it would press this in. By having openings in the throat region, it allows us to equalize the pressure on both sides of the tympanic membrane and allow it to maintain free movement really with those sound waves. So from there we go into the inner ear. The inner ear, this is housed within the temporal bone. What you're going to have is you have these, pretty much have a bony cavity lined with some epithelium that is called the Boney Labyrinth. And it is a fluid-filled space. Within this Boney Labyrinth, there's also going to be a membrane that's traveling up through the middle of it called the membranous labyrinth. The Boney Labyrinth has fluid surrounding it, which is surrounding that membranous labyrinth, and then within the membranous labyrinth, you're going to have something called, you're going to have that perilymph inside the membranous labyrinth. But you have this endolymph surrounding the membranous labyrinth on the outside. Perilymph is, it's the endolymph that's inside the membranous labyrinth. You have perilymph surrounding the outside of that membranous labyrinth and helps hold it in place. And what's going to happen is these fluids can actually transmit when you get that stapes pushing in and out due to those vibrations. It's going to create pressure waves within this fluid, and that is how we're going to activate our different receptors for hearing. So when we look at this Boney Labyrinth, it's actually a combination of three different things. You have the cochlea, which is where we're going to be dealing with sound. You have the vestibule, which is this combination of two sac-like structures called the utricle and the saccule, which it seems like I'm making these words up, but this is what they are called. That is where you have the static equilibrium type of things, the head position type of receptors. And then we have what are called the semicircular canals, which are three different tubes in the different X, Y, and Z planes that are going to help with rotational movements of the body. So you can kind of see that here. So again, this is kind of showing you that membranous labyrinth housed within this bony labyrinth here. You can see it right here really well. So you can see this would be the Boney Labyrinth on the outside. You have this little hunk of membrane that's traveling through the center of it. So Boney Labyrinth out here, membranous labyrinth here, perilymph, endolymph. And you can see this is that cochlea that's spiraled up like this. You can see the utricle and the saccule, which are part of that vestibule at right angles to one another, and then you can see the 3 semicircular canals. With the utricle and saccule, as well as the semicircular canals, we will come back to those when we do the vestibular senses and talk a little bit more about what's going on there. What we're really worried about for sound is going to be the cochlea. So it is a snail-shaped one here. It houses what we call the spiral organ, which is that kind of Boney Labyrinth surrounding the membranous labyrinth, traveling up there. All together we call it the cochlear duct. So this is kind of showing this idea right here. This is showing it kind of lengthened out, but you can see if it's spiraled up here, you can see how there's going to be a fluid-filled cavity along the top called the scala vestibuli. When we get pressure waves from that stapes, it's going to cause those pressure waves to travel out to the tip and then work their way back, and then this round window can let those pressure waves kind of go in and out. This is this cochlear duct, and when they kind of get all spiraled up like this and you slice through it, that's what they refer to as that spiral organ. So this is where you're going to have all these sensory cells, which are again going to be what we call hair cells. They're not actually hairs. These are going to be specialized types of almost like a cilia or a villus that are going to have different receptors that can respond to mechanical vibrations. So these hair cells are what are going to have these sensory receptors, and when these are activated, they release neurotransmitters to the neurons, which will take that information back to the brain. And like I said, these hairs cells are really what is looking like hairs are these really long microvilli, which are referred to as stereocilia. And we're going to have this little gelatinous membrane that the stereocilia kind of push up into, something like this, and that is called the tectorial membrane, and we'll look at all this in just a second here. So you can see here is that spiral organ. You can see the scala vestibuli, scala tympana going back. You have the membranous labyrinth right here. This little tongue-shaped structure right here is referred to as the tectorial membrane, and you can see you have all these hair cells that are going up and are in contact with that. When we get vibrations that move through these scala vestibuli and scala tympani at certain spots, it's going to cause this basilar membrane to push these hair cells up into this tectorial membrane, and that bending of those stereocilia are going to be what leads to action potentials being sent to the brain and leads to us perceiving sound. So they talk about this upper membrane right here being the vestibular membrane. This is down here is that basilar membrane, and this whole structure right here is this cochlear duct. And really what we're worried about in this cochlear duct, where all the sound reception is going to take place, is what we refer to as this organ of Corti, which is the hair cells and the membranes above and below that. So if we look in at this a little bit more, this is where these hearing receptors are going to be. So you can see this is the basilar membrane right here with hair cells above it. Those hair cells are going up and touching this tectorial membrane. And again, any deformation down here or squishing in from the top can cause this tectorial membrane to move against these hair cells, bend those hair cells, and trigger an action potential to be sent to the brain from these hair cells. Again, depending on which hair cells we bend, where it is found within that cochlea is going to give us our different sound frequencies, our different pitches, but that's going to lead to a nerve impulse being generated and sent back to the brain. And here's what you can see in real light micrograph, so an actual image from a slide. Shows you this tectorial membrane, the cochlear duct, and those hair cells. And again, this is why they're kind of referred to as a spiral organ. It kind of looks like a really crazy looking tree, but you can see how this spirals up here. It's an easier way for all the nerves to get into these areas here and not have them wrapping around everything. So the structure actually does make some sense when it's spiraled around like that. A lot of times when you see that snail shell, it's like, why would we do that? Really because it allows this main nerve to travel up and get to all these hair cells without being some outrageously large structure. It's actually a pretty efficient way to get a lot of nerves to these areas. And this is the natural micrograph of it. Again, I'm going to have you know a little bit about the anatomy of this one. We'll do a little bit of that on the next quiz. But again, knowing the organ of Corti and the different parts of that, I would say that's stuff that would be definitely fair game. All these structures, not necessarily. But again, the scala vestibuli, the scala tympani, the vestibular and tectorial and basilar membranes, where those hair cells are at, those are all things I think are good things to be aware of and understand. And again, it shows you in the cartoon version here how those hair cells are up into that. And again, when this structure gets moved against the stereocilia, that is what's going to lead to a change in the membrane potential of these cells and lead to something being sent down the cochlear nerve back to the brain where we can actually perceive a sound. So to kind of give you the basics of how this works, we're going to have sound waves that are starting out here. They're going to go and be directed into the auditory canal by the auricle. Those sound waves are vibrations in the air, and because the tympanic membrane is much larger than the oval window, and we have a direct mechanical connection between those ones, we can generate a lot of movement in that oval window by the fact that the tympanic membrane is much larger. So it is going to amplify those vibrations a little bit, and as that tympanic membrane vibrates back and forth, it's going to move the malleus, which moves the incus, which moves the stapes. That stapes is going to push in and out on the oval window, and again, because the tympanic membrane is much larger, it amplifies those vibrations in there. And what this, by pushing in and out on that oval window, it's basically kind of acting as a piston, which is going to push pressure waves at different frequencies, depending on the frequency of the vibration, through that fluid in there, that perilymph. So it pushes in on that perilymph. That's going to be transmitted down the scala vestibuli to back to the scala tympani, which is going to cause the round window to move in and out on the other end. Depending on where the frequency is, what the frequency is, it's going to deform the walls of these scala vestibuli and scala tympani and press in on the endolymph and the cochlear duct. And when it presses that endolymph in the cochlear duct, it's going to cause that basilar membrane to move against the tectorial membrane. And when that tectorial membrane bends those hair cells, that's going to lead to a change in membrane potential within these ones, which can generate a nerve impulse going back to the brain. And this is kind of showing you how that happens. You have these vibrations going here. It's moving this tympanic membrane, which transfers these through here. This presses in and out, which sends pressure waves through here. Depending on their wavelength, i.e. frequency, it's going to deform this somewhere and cause those hair cells to get pushed against the tectorial membrane at somewhere along here. What we generally see is higher frequencies are going to be closer to the window end. So higher frequencies will cause the formation up here. Lower frequencies tend to travel out more towards the tip before they deform this membrane. So depending on which hair cells you activate along this cochlear duct, that is going to give you those different pitches of sound. Again, amplitude is more of the size of the wave, how big the vibration is. That is not going to affect where we do it along the cochlea, along this cochlear duct here, but it may activate more hair cells, which can give us some type of measure of volume. So like I said, pitch is the frequency of those vibrations. So something that's low like that would be low pitch. Something that's high like that would be higher pitch. Again, higher frequencies, closer to the end with the windows, closer to the beginning where the stapes is. Lower frequencies tend to travel out more towards the tip of that. And we can hear anywhere from about 20 hertz to 20,000 hertz within our hearing range. Obviously, as you age, you start to lose out on the low end and the high end of this a little bit more. So, and again, this is kind of showing you those high frequency ones closer to the oval end. Medium frequency, some in the middle, and the lower frequency are going to get further along before it deforms that membrane. Again, depending on which hair cells are activated, that's going to give you that different perception of pitch. Intensity or volume is kind of more of the amplitude of this. So obviously if we get a bigger vibration, if you think about waves, you can have waves coming in at the same frequency. So a wave comes into shore every so often. Every once in a while you could get a bigger wave. That is amplitude. If we get a bigger wave or a larger vibration, that's going to be a louder sound. So you get a bigger wave here. One of the things it can do is it can activate more hair cells in that area, which would be one way we would determine that this is a louder sound. The other thing we're going to have is you can have certain hair cells that have a different threshold, so it needs a bigger movement. So we need a bigger vibration to activate those hair cells. And that's where we can get some of this differentiation in volume. And again, we can have a lot of levels of difference that we can understand, be anywhere from a whisper to really loud noises. Generally, we measure sound intensity in decibels, which is an increase in intensity of 1.26 times, which is a really weird unit. But this kind of gives you an idea. I'm probably talking at about 50 decibels right now. You can see some of these ones here. And again, as you start getting these louder decibel ones, these hair cells are fragile. Loud sounds can vibrate them to the point it damages those. Certain things here can create such a big pressure wave they can blow out the tympanic membrane. So things like a gunshot, jet planes getting too close to you can actually rupture that tympanic membrane, which would stop you from being able to hear those sounds. We do have muscles that can hold on to these ossicles and tension them a little bit, and this is a way we can tone down louder vibrations or louder sounds. So we can actually reduce the sounds a little bit by about 30 or 40 decibels, but it's not a particular fast reflex. So if you get an immediate loud sound, it can cause damage to the hearing. The other thing is when we start thinking about senses, a lot of times you can lose a sense, and deafness is the loss of our sense of hearing. What's interesting about deafness, a lot of our other ones, it's either you have the sense or you don't. Generally, on almost all these ones, you can be damage to the receptor, damage to the nerve that's taking it back to the brain, or damage to the brain region that decodes it. Those are all ways you can lose a sense, like sight. That's also true of deafness. The one thing that's different with deafness is because we are transmitting sound waves from outside of our body through that outer and middle ear to the inner ear. If something damages those mechanisms that transfer those sound waves mechanically to the inner ear, that can also lead to deafness. And when you don't have that, that's referred to as conduction deafness. You've lost the ability to conduct these vibrations. Obviously, you put an earplug in and go like this, that would be conduction deafness. If you get too much earwax, the otoliths, those little bones in there get damaged. If you destroy the tympanic membrane, those are all ways that you could have conduction deafness. Nerve deafness is damage to kind of the receptors, the nerve taking it back to the brain, or the brain region. Those would all be things that we would refer to as nerve deafness. A lot of times, low frequency and high frequency sounds have a tendency to cause loss of being able to perceive those pitches. Loud sounds tend to get rid of your low frequency, and again, a little bit of the high frequency too. Then there is certain antibiotics that have caused damage to the hearing kind of across all frequencies. Every once in a while, you can have a bad reaction to that. So that gives us kind of the basics of how this is working. So again, sound waves being transmitted from out here through those different parts of the outer and middle ear to the inner ear, and again, it's going to be that deformation that causes those hair cells to go against the tectorial membrane, which is going to cause a change in membrane potential. I'm not going to get super deep at all into any more than that. That is really the basics of how we hear sound. Again, if you were to actually go with the nitty-gritty within that cell, the hair cells, it is going to be similar to what we saw with nerve cells and other things like that. You get enough of a stimulus, it causes enough of a change in the membrane potential that reaches a threshold and sends a message. So I will see you next time.